really looking forward to hearing all about the kind of progress on the Pacific project. So I'll hand over to you both.
That's great. Thank you very much, Joe. Right, I will just share my screen. Hopefully that will come through. Joe, can I just check that that's visible to you?
Yes, it's, it is, yeah.
That's great, thank you. Okay, so yeah, thank you very much for the introduction. So as Joe said, I'm going to be talking about the NERC Pacific project. This is going to be a double act between myself and Amy Thorpe. Amy is a researcher at UKCEH. And I'll just start off with a very quick high-level introduction for the Pacific project, but what we're not going to do is do a sort of blow-by-blow account of all the different work packages. We're focusing on one particular theme just to share with you some of the early results and publications that are coming out of the project. But this is very much a collaborative project. It's working with 4 organisations. So the project's led by myself at UKCEH, working with my colleagues there across a range of disciplines. But we're also working with Barbara Kasprzyk-Horden at the University of Bath and her team, Michelle Jackson and her team at the University of Oxford, and Kerry Walsh and her team at the Environment Agency. And we have interacted and worked with a wide range of different stakeholder partners during the course of the project. So what we're planning to cover today, I'm just going to give a sort of general introduction for the sort of rationale for the project. So a brief introduction to freshwater ecosystem health, the historical trends in England, current status, and a bit of a focus on future threats that sets the scene as to why we're doing this research. I'll spend a bit of time talking about bioindicators, so how we measure the ecological health of freshwaters, talking a little bit about the traditional approaches and some of their limitations that are the rationale for why we're using microbes to look at the health of freshwaters. I'm then going to pass over to Amy, who's going to talk about a couple of our early publications and data, which is building on a very large national England scale data set in collaboration with the Environment Agency from a big programme they have called the River Surveillance Network. I'll talk a little bit about some additional work we've done around microbial indicators and give you a bit of a sight for some of the future work we're doing on this project. So I'm just going to start off by briefly introducing the context for the research. And I'm sure you're all very aware that freshwater ecosystem health has had a huge amount of focus over the last probably sort of 5 to 10 years. It's been reflected in conversation on social media, in the traditional media. So I've just got some snippets from online newspapers, and it's become a major topic of political discourse. And in my view, this has been driven by 2 major factors. I think the first has been the increased visibility of data, particularly data on combined sewer overflows, which has brought to the public attention potential pathways to pollution into freshwaters. And then I think the second thing is the increased interaction from the general public with freshwaters. And, you know, I've seen this over the last 10 to 15 years. I live in Oxfordshire, not far from the Thames, and there's been a huge increase in the number of people swimming in rivers, kayaking, and particularly paddleboarding. So I think this has really raised the awareness of the general public around the health of freshwaters and making them care, which is a fantastic thing. But it is worth noting that actually the health of freshwaters generally has improved over the last 4 decades. So this is just some data here I'm presenting from the Environment Agency. So this is about, I think, 135 river sites, the harmonised monitoring scheme that's run for the last 4 decades. And you can see for major sort of traditional measures of pollution, such as ammonia, biological oxygen demand, which is a measure of organic pollution, and nitrate, these all peaked around 1990 and then have shown significant declines in the subsequent decades. This is also true for orthophosphate, again, a nutrient pollutant that comes from both wastewater and agricultural sources, peaked in the mid-90s and then has declined. The only one that really bucks the trend is nitrate. This is a pollutant that moves very slowly through ecosystems. It persists in soils, it persists in the subsurface. So even if we stopped all inputs of nitrate into the freshwater system, it would take a very long time to move out. But alongside this, there has actually been improvements in biodiversity as well. So there's been a lot of work on large Environment Agency data sets on macroinvertebrates, one of the key taxa that are used for looking at the health of freshwater ecosystems, as I'll talk about in a minute. And a number of different studies have looked at these data and showed that there's been general increases in the number of family or different taxa that are found over this period of time, which makes sense because it's been linked to the overall improving water quality. However, it's worth noting that these national trends definitely don't cover all rivers, they don't cover all subcatchments, and they don't cover all specific parts of the rivers. So it's entirely plausible that even though we have this national background improvement, that there are specific problems associated with particular locations. The other really important issue to note is that the nature of pressures in freshwaters is changing. So in the past, it was primarily associated with nutrient pollution, metal pollution, but there's increasingly a wide range of manufactured chemicals and substances that make their way into freshwaters. And I've just got some examples of these emerging contaminants on the left-hand side here. So classes of organic compounds that we use on ourselves in the form of medicines. We use them as personal care products in shampoos and sunblock. Industrial chemicals that make their way into food, into packaging material, household products of a wide range produce chemicals that end up going down the drain. Metals are still a significant problem because they're used in a wide range of different processes. And there's been an increased awareness around particulate pollutants, so particles that come from tyres, that come from brakes, that come from larger plastic packaging that break down into smaller and smaller fragments. So there's been quite a lot of work on this that have shown that these wide range of chemicals can show a potential threat to the health of freshwaters. So this on the right-hand side is an example of a continental scale modelling exercise where they really showed that this is related to population density and the capacity of the environment to dilute the contaminants that we put in it. But the other major pressure, and this is really relevant at the moment actually, is that our climate is changing. And this has significant impacts on freshwaters as well. So droughts reduce the availability of water, so they reduce the ability of the environment to dilute pollutants that come into it. Heat waves, which is a very relevant topic, put heat stress on the environment and interact with organisms in terms of their ability to respond to other stresses. And floods mobilize pollutants into freshwater environments. So whilst there has been an overall increase in the ecological quality and the chemical quality of rivers, there are new and emerging threats that we need to be aware of. So how do we go about measuring the health of freshwater ecosystems? And this has been traditionally done by looking at groups of organisms and having a good understanding of their natural history. So what are they? Where are they found and how do they respond to environmental drivers? And this builds a picture of ecosystem health. So groups of organisms that have traditionally been used to do this include fish, macroinvertebrates, macrophytes, so aquatic plants, and a particular group of algae, of microbes, the algae, particularly the diatoms, which have all been used, and we have a good understanding of these organisms and how they respond to a range of pressures. And the sort of core principle behind this is that once you understand what species are there, where they are, and how they respond to the environment, you can start classifying them as either sensitive species. So these are ones that they don't like pollution, so you tend to find them in high quality environments, or tolerant species, so ones that are more tolerant of high levels of pollution, and we tend to find them in more degraded environments. And by having a good understanding of these species and how they respond to these different drivers, you can use what species you find in a particular environment to tell you something about the health of that ecosystem. However, there are limitations with these more traditional methods. So some of them don't have sensitivity, so they're not always very sensitive to particular stresses. And as I've already said, these stresses are growing more complex over time. The specificity may not be very good, so they may be responding to a stressor, but you don't know what the stressor is. Many of these are quite variable, so depends what time of the year you sample or where you might sample in a river catchment will give you different data. And there's also just a practicality side of it. So particularly for species like fish, they are mobile. Surveys can be quite expensive and time consuming to get relatively small amounts of data. So because of that, there is a need for alternative approaches that might support the way in which we actually manage and understand freshwater ecosystem health. So as you probably will have already guessed from the title of this talk, we're interested in microbes, and I'm calling them the hidden majority because they are the most abundant organisms that you find within the environment. So if we look at a water sample, they may have a single drop of water, so 1 milliliter will have anywhere between 1 and 10 million cells per milliliter, representing a huge diversity of organisms. This is the same for microbes that grow on surfaces of materials. So every surface within a freshwater environment is covered in a thin film of microbes. So on the surfaces of plants, on the surfaces of the substrates in which are saturated with water, and on the surfaces of rocks, these so-called epilithic biofilms, which is the picture on the left-hand side. And again, these are incredibly populous. So there's anywhere between 1 to 100 million cells per square centimetre, representing a huge diversity of organisms. And this is just a sort of sneak preview of some of the data we've been generating as part of this project. But a single freshwater biofilm sample, so this is one growing on the surface of a rock, will contain anywhere between, well, around 720, I said species, but what we really mean is different types, because species concepts in microbes is a bit more complex. But around 720 different types of bacteria, 170 different types of fungi, 260 other types of microbial eukaryotes, and a currently uncharacterized and unknown diversity of viruses. So these are the most abundant organisms we find in freshwaters, but they are by far the most diverse as well. And they're not just abundant and diverse, but they are performing really important ecological roles within freshwaters. So microbial communities are responsible for controlling the turnover of organic material, the nutrient cycling within freshwaters. So the cycling of nitrogen, the cycling of phosphorus, organic matter, carbon. They control greenhouse gas fluxes, which are really significant at a global scale coming from freshwaters. So these microbes, they're not just potentially a useful tool, but it's important to understand what they're doing and how they're responding to these environmental drivers to be able to understand how our freshwaters may change in future. So the core principle of the project really is to understand freshwater ecosystem health from a microbial perspective. And the core premise or the hypothesis of this work is that a healthy freshwater environment will have a healthy microbiome. It will contain a natural array of species performing functions as they've ecologically evolved to do. But in a more degraded environment, so maybe one that's stressed by changing climate, one that's stressed by contaminants coming from a range of sources, we may expect to see a microbial community that's disturbed by these different pressures and drivers, and that may have impacts on the functions that they're performing for freshwater ecosystems. So the core sort of premise of the Pacific Project is to try and understand, can we unpick these signals in the microbial communities, and can we use them as tools to better understand the health of freshwater ecosystems? So if we want to generate new bioindicators, microbial bioindicators, we need to understand who's there, we need to understand the taxonomy, we need to understand where they are, the biogeography, and we need to understand the environmental variables that they respond to, so their ecology. And really they're the sort of core principles of building a new bioindicator suite. So the final thing I'll just briefly talk about before handing over to Amy is that identifying microbes is quite complex. It's actually the reason why they're the sort of hidden majority and relatively unstudied is because they are hard to study. They're difficult to grow in the lab. They're by their very nature very small. They're morphologically conserved, so they look very similar to one another. And as I've already said, they are incredibly diverse, hundreds of species per sample, that makes them very challenging. So because of that, we use molecular methods, so the analysis of DNA to study microbial communities. And the core principle behind this is we can take a sample, in this case a biofilm sample, we can extract the DNA, we can prepare it for sequencing on a platform called a high throughput sequencer. These will generate anywhere between tens and hundreds of millions of sequences per sequencing run. And we have two core approaches available to us. The first one is called amplicon sequencing. So this is a method by which we look at a particular gene, and the variation in that gene between different species can enable us to identify the members of the community in that sample. So it enables us to identify who is there, what species are in my sample. There's also a more complex and expensive method that we can use called metagenomic sequencing. And this enables us to not just look at who's there, but we can look at genes associated with all those biogeochemical functions I was talking about. So it's who's there and what are they doing. And hopefully this will nicely prime you for some of the work that Amy's been leading, where she's going to talk about some recent papers we've published on looking at who's there and what they're doing at a national scale. So Amy, I'm going to pass over to you if you want to take control.
Thanks, Dan. So yes, these next few slides are going to go through some of the results from a collaboration we've been working on under Pacific with the Environment Agency. So this is the first national scale study of river biofilms in England. And so the Environment Agency have been collecting biofilm samples from across their river surveillance network. And this is a series of sites across England that they routinely monitor to track long-term changes in river health. And these are all these blue sites in this map of England here. And these samples are paired with really detailed environmental data. So this includes water chemistry, catchment land cover, and geology. And so for over 1,600 of these biofilm samples, we've extracted DNA and done metabarcoding or amplicon sequencing for 4 different markers to investigate the diversity of bacteria, microbial eukaryotes, and also more targeted amplicons to look at fungi and diatoms in more detail. And then for a subset of 450 of these samples, we've also done metagenomic sequencing to look at the functional diversity of these communities. So this involves assembling these short sequences of DNA into these microbial genomes, or they're also known as metagenome assembled genomes, or MAGs. And we use various different databases to annotate these genes with their functions. So the first few slides I'll go through are looking at the bacterial community using the metabarcoding data. And so we've detected a huge diversity of freshwater bacteria across the river network. And this is recovered over 24,000 bacterial ASVs, and these are amplicon sequence variants, so essentially unique sequence reads that are kind of a step further beyond species. And these were assigned into 1,000 unique genera as well. And so this plot shows the relative abundance of these different bacterial groups across one of our samples, and these are arranged by sampling date, so this is season and also year. So we've been running this from 2021 to 2023. We've also got 2024 samples as well, more recently. And we can see that the community is really dominated by a few groups. So this is Bacteroidota, which are these dark green band near the top. Cyanobacteria, which is the lighter green, which show a slightly more variable abundance across the time series. And also Pseudomonadota, which is the big band of blue. And these are accompanied with a rarer composition of bacteria. So these are kind of the purples and the orangey colours that you just see small bands of. And this is Nitrospirota, Planktomycetes, and a few other groups. And this community composition is relatively stable year to year and even across seasons. But we do see that some of the rarer groups appear more in the autumn, while we've got like cyanobacteria, which are more dominant in the spring. And this is really consistent with the occurrence of spring blooms when temperatures and nutrients tend to be higher. And this seasonal pattern is further illustrated by the slightly higher alpha diversity and richness that we see in the autumn compared to spring. So we found that biofilms are often dominated by a smaller number of abundant taxa in spring. And then in autumn, we're seeing more of those rarer taxa. And looking at the ordination on the right-hand side, each point on here represents an individual sample, and samples with more similar bacterial communities get plotted closer together. And so we see a slight separation between the spring samples in green and the blue samples, which are the autumn samples. But there's still quite a lot of overlap between these. So this suggests that while seasonality does play a part in structuring these communities, it's not the dominant driver explaining the differences we're seeing between our samples. And we've instead found that the physiochemical drivers within the water column were more influential in shaping bacterial community composition and diversity. So we found that alkalinity was the strongest factor shaping the communities, and this is likely reflective of the underlying geology of a river's catchment. So limestone or chalk catchments tend to have a higher alkalinity, and this affects the river's buffering capacity and ability to resist changes in pH. And we know from work in soils that pH is really an important driver of bacterial communities. And this was followed by dissolved oxygen, nitrate concentrations, and also temperature. But we found that other factors such as orthophosphate and dissolved organic carbon concentrations explained a relatively smaller proportion of the variation. And we've also found that samples towards the bottom right of each of these plots, so if you can see my mouse, it's kind of these ones in this bottom quarter. These are slightly more distinct from the main cluster of samples, suggesting that they have quite unique communities. And we can see from the colour gradients of each of these 4 plots that they have quite a distinct water chemistry as well. So they tend to have lower alkalinity, higher dissolved oxygen, really noticeable lower nitrate concentrations, and generally lower temperatures. And from looking at some of the land cover data, we can see that these sites were mostly the upland acid grassland sites, which are typically more pristine, so they could represent a cluster of samples that are less impacted by anthropogenic activities. And so we're using this data to identify potential bacterial indicators of ecosystem health. So using the relative abundance of the bacterial taxa alongside environmental drivers, we can identify points along an environmental gradient at which communities start to respond and change, and which taxa are the effective indicators of these changes. So each of these peaks represents the point along a pressure gradient, such as nitrate concentrations or temperature, at which we detect a shift in the relative abundance of an individual bacterial species. Some are tolerant of increasing pressures and may become more abundant as they increase. So these are each of these red peaks here. And other ones are much more sensitive to these pressures and may decline in response. And then these dashed lines show the average community level change point. So this represents the point along the environmental gradient at which overall community composition change is the greatest. And these taxa highlighted here may represent promising taxa that can be used as bioindicators, and their thresholds or change points can help us to understand how an increase or decrease in a certain environmental variable may influence microbial diversity and ecosystem functions. And then moving on to some of the findings from the metagenomic data, where we're linking community composition to function. So across the 450 biofilm samples that we performed metagenomic sequencing on, we recovered over 1,000 bacterial genomes, which span a really diverse range of bacterial phyla shown in this phylogenetic tree. And we also found that a significant proportion of these genomes could represent novel taxa. So these are bacteria that may not have been described before. So this bar plot shows the proportion of genomes at each of the 4 taxonomic ranks that are represented in the taxonomy databases in blue, and those that are potentially novel in grey. And we found that just over 95% of the genomes at the species level may be novel, and around 20% at the genre level. And so these novel bacteria may not have been previously described, and it represents many species of bacteria living in river biofilms that we don't know much about. And so we found that some of these biofilm bacteria are involved in a diversity of functions, which span the uptake and degradation of organic compounds, different trophic strategies, and responses to stress, which really highlights their functional diversity. So some of these bacterial taxa, such as Pseudomonodonta and cyanobacteria in particular, each have a very wide diversity of functional roles and metabolic traits. And some of the functions that are common across nearly all biofilm bacteria were genes involved in biofilm formation. So that's like the production of the EPS or extracellular polysaccharides that kind of sticks the microbes together in the biofilms. And that's to be expected because we're looking at biofilms, but there was also quite an array of genes involved in the transport of metals and organic pollutants, and this suggests that they may have a role in degrading these pollutants in rivers. And then just focusing on some of the genes involved in nutrient cycling. So we've reconstructed the pathways for carbon cycling in blue, nitrogen cycling in green, and sulfur cycling in orange. And each of these steps is annotated with the number of genomes which had genes encoding that step. And we found that virtually every step in these pathways are really well represented by the biofilm bacteria. And this really illustrates their importance in nutrient cycling and their foundational role in making nutrients available to higher trophic levels. And I'll hand back to you, Dan.
Thank you, Amy. Okay, so I'm just going to try and quickly wrap things up, just giving one more example actually of where we've started to try and identify indicator taxa building on Amy's work. So this was some work led by John O'Warren at the Environment Agency. And what he wanted to do was identify, particularly look at taxa that respond to nitrogen pollution. Again, this is building on this very large, fantastic data set we have, nearly 1,600 samples, nearly 700 sites, covering a huge range of nitrogen concentrations, which is represented on this histogram on the right-hand side. So from very clean sites, barely measurable 0.001 milligrams per litre up to around 32 milligrams per litre of nitrate. So we have a big range of screens of concentrations, sorry. And what John has been doing is, gone ahead. John has been using machine learning to try and identify taxa that respond to nitrate concentrations. So he's used a couple of different machine learning approaches, one called random forest and one called XGBoost. These are methods that can be used to identify taxa that will respond. And then he's coupled this with an additional method called threshold indicator analysis, which is what Amy's used to identify concentrations at which the community respond to. So this has been done at the genera level, just under 700 genera. It's identified about 193 predictive indicators and split these into indicators that are tolerant of pollution and ones that are sensitive to nitrogen pollution. And from this, we can build predictive models. So both of these graphs on the top left, they represent the results from XGBoost. And this shows, looking at this core set of taxa that we've identified will respond to nitrogen, we can build a pretty good predictive model. It's not perfect. If it was perfect, it would match along that one-to-one line. But just from this sort of smaller subset of taxa, we can predict nitrate and total oxidized nitrogen pretty well. And we can also use these additional analyses. This is called a SHAP plot that enables us to identify genera that have high predictive value. So these shows which type of bacteria are useful for predicting nitrate levels in water. So we're starting to move towards this better understanding of the natural history of what's there, which species are there, what functions they're doing, and then how they're responding to particular drivers. The other thing I just briefly want to highlight, and this is really sort of looking forward as to the work we're now doing. All of the work we've showed so far has been looking at bacteria. This graph on the right-hand side shows the diversity of these different groups of organisms we're looking at. Bacteria and archaea represent the most diverse, around sort of 700 odd per samples, but up to around 1,500. But we also have fungi, we have microbial eukaryotes, and then we also have an assay that looks specifically at diatoms and a few other species of algae. And these represent a sort of incredible diversity of organisms. So this tree in the center represents, each branch represents a different group of microbial eukaryotes. So this is looking at this microbial eukaryote assay. And from within this, we can identify golden algae, diatoms, ciliates, cercozoa, green algae. We do get some terrestrial plant DNA, likely sort of shed from the environment and captured in these biofilms. And we also get some fungi as well. So there is a huge, as I've said before, hidden biodiversity in these samples that we can use to start to explore what's there, but particularly how they're responding to different environmental drivers. And this is just similar to what Amy did. So these are ordination plots. Each point represents a single sample, a community of eukaryotic organisms. And again, we can see that actually there are clear environmental drivers that start structuring these communities. So the next step for us is actually to start delving into this more, using some of these machine learning techniques to identify which taxa respond to which drivers, and can we build these predictive models that will actually potentially be operationalizable for understanding and managing the environment. The final thing I just briefly want to talk about before we finish is that in terms of functions, it's not just about functions involved in biogeochemical cycling. Microbes have a whole array of other functions within them. And this is some work that's been led by Martin Spur at the Environment Agency to look at antimicrobial resistance. So this is a class of genes involved in resistance to chemicals. In the case of antimicrobials, it's the medicines we use to treat microbial infections. Now, the interesting thing about these is they can be diagnostic for chemical pollution in the environment. At least that was our theory. So what Martin's done is look at these metagenomes, identify copies of genes associated with resistance to antimicrobials, and we've been able to map this out, and you can see there are sort of hotspots across different parts of the country. The southwest and the northwest, you can see tends to be slightly higher. But what he started to do now, and I should say this is a paper that's currently in review and undergoing revisions, so these are preliminary results I'm just presenting here, is that we've started to look at loads of antimicrobial resistance in related to potential sort of obvious drivers, such as how much wastewater. So all our samples into 3 main clusters: a low sewage cluster, a medium sewage cluster, and a high sewage cluster, and then looked at whether or not we can pick up antimicrobial resistance genes that might be associated with the input of wastewater into the environment. And long story short, yes, we can. We can pick a class of genes. So this heat map on the right-hand side shows the abundance of particular genes relative to this low wastewater input. And we can see there are some diagnostic antimicrobial resistance genes involved with virulence, resistance to sulfonamides, and a range of other antimicrobials that look to be diagnostic of wastewater pollution in the environment. So this is some work we're actively progressing at the moment, and there should be hopefully a paper coming out on this relatively soon. So on to the conclusions. Well, I think hopefully we've shown to you that freshwater microbiomes represent this hidden biodiversity of really quite understudied organisms. They are really functionally important. They play critical roles in nutrient cycling, the flux of greenhouse gases, the overall health of freshwater ecosystems. They respond to physicochemical drivers in predictable ways that make them suitable as sensitive bioindicators. And then finally, there are a whole array of other functional genes that have the potential to serve as indicators of anthropogenic pollution. And we're really just starting to scratch the surface for how we can interrogate this data to potentially build useful tools for environmental management. So final thing is just thanks. This is just part of the Pacific project. There are other lines going on where we've done some experimental work, both in the lab and the field. There's a modelling component, and maybe we'll talk about some of these in a future webinar. But yeah, it's very much a collaborative project. So a huge thank you to all of those involved with the project, and thank you to all of you for listening. I'll be happy to take any questions.